organisms develop through a series of stages, leading to sexually mature adults. In a few cases, ontology reversal is possible, but it does not occur typically after the onset of sexual reproduction.
But you know what? There is an exception. Hey guys, welcome back to Philosophical Enterprise. This is your Geek Goddess, and today I'm here to answer all of your questions. At the second conference of the Hydrogen Society, a paper was published. It was so revolutionary that the members of the audience were so incredulous that they were not even willing to believe it was true. One of the attendees, who was late Victor Skemeb, was so doubtful about that paper that he literally said it was impossible. This all started with an observation which was purely by accident. In the late 1980s, two laboratory German students, Giorgio Bevistrello and Christian Sommer, they just basically took a specimen of a jellyfish which was Totopsis nutricula. It is basically a jellyfish which is less than a centimeter long. This small predatory invertebrate has mainly two stages in its life cycle. First of all, it is a polyp forming colony, which is a group or a colony of polyps which is in the bottom of the sea, if we just say. It is a fixed form which is called hydroid, as it is a collection of small forms of the smaller or younger jellyfishes, which is polyp. It's like a plant and a stem, like hydra. The structure is like that. It's not exactly like hydra. And the second one is a more recognizable jellyfish form, which is the sexually matured form known as the medusa. So basically, the individuals which were collected were believed that they were the immature medusa form of the nutricula. So as a basic observation process, a typical observation process for the life cycle of the Totopsis nutricula or a typical jellyfish, the students just took the specimen in the laboratory and reared it. After being collected, they basically just reared it in the lab, as I said, and the Totopsis specimen, the hydroid, also released the medusa form, and after that the students just forgot about it. But afterwards, when they returned, they believed and they just literally expected that there must be a adult medusa form, because the time had passed and now it's time for the jellyfish to be a sexually matured adult. So, but there's the catch. What happens? It is a plot twist. Normally, in a hydrogen life cycle, immature medusa form or not so mature, sexually matured medusa form matures and then just spawns eggs and sperms, as it is a sexually matured medusa. And when this sperm and egg fertilize and forms an egg, then this larval form is known as a planula. When these planula settle somewhere, then they just form a hydroid colony, a new hydroid colony, which has many polyps, and these polyps release new medusa, and this life cycle just goes on, and the adult medusa dies. Now, if I talk about this case, the medusa of the species that they believe to have collected were not having matured gonads. They thought that so this immature medusa form has to just grow and mature and just spawn that sperm and ovum after maturing and being a more sexually mature medusa, eventually producing planulas, which is a kind of larval or fertilized egg type thing. So we have basically discussed it, but what happens in this particular case, like in our Totopsis nutricula case, that they thought that it was nutricula. There is a catch. So basically they knew that it was an immature form of the species. It has to grow up and become more sexually adult, a medusa form, and eventually just release that fertilized egg type thing or the planula. And this planula normally just goes down, as I just discussed, and just becomes a hydroid, and this hydroid releases medusa form. Normal life journey, right? And again and again, when these planula settle down, then they create more polyp colonies, which are basically hydroid. Polyp colony is known as a hydroid. So as a result, more medusas and more planulas. So eventually, this sexually adult or mature medusa dies, like because of age in the Nutricula species. Like what they observe is basically, instead of finding all that typical thing, they literally observed less adult medusas as compared to the earlier, and they just found more babies, lots and lots of babies in the newer and younger polyforms in the bottom, and there were less adults, and they were just thinking, what happened to these adults then, right? And of course, it was not possible for these medusas to reproduce in such a short term of time. And if they even reproduce, okay, fine, they just reproduce in a quick way. So what happened to those adults then? Even there were not even other specimens of the body at the bottom. So basically, after all this confusion, to find out the answers of these questions, the researchers basically started to keep a look at the individuals present in the tank. Okay, and what they found shocked them. The adult medusa forms were not spawning and reproducing to form younger polyforms. They were literally reverting themselves back to the juvenile form, completely reversing the aging process. So with this, we can say that the polyps in the bottom of the tank were not new individuals, but the sexually mature medusa who changed itself into sexually immature form, or the baby polyform. How is it even possible? What they realized afterwards was that what they have collected was not Teratopsis nutricula, but another jellyfish called Teratopsis dohrnii. One they realized that it was capable for performing Benjamin buttoning effect over and over again, to the point scientists has found something inconceivable: immortality. So to begin with, what really is a Teratopsis dohrnii and how is it really different from other jellies? The immortal jellyfish is not a Scyphozoa, which is a basic scientific name for a true jellyfish, which is the most common class of a jellyfish, Scyphozoa. But on the other hand, it is a Hydrozoa, which is a jellyfish basically lives more of its lifespan in its hydroid form, which is that that colony of polyps which is settled, and it releases medusa, as I told you earlier. But of course, these both classes of the jellyfish, Scyphozoa and Hydrozoa, belong to one specific phylum, which is phylum Nidiria. Now here is a point of note. Basically, we use the term jellyfish for Scyphozoa, which is a true jellyfish, but for our familiarity, I'm also going to use the term jellyfish to call out our Hydrozoa, which is Teratopsis dohrnii. Now, how was it discovered? So Teratopsis dohrnii, which is less than the size of a full-grown pinky finger of a human, in the length, was discovered in the late 1883s, in the Mediterranean Sea. But its immortality, its ability to be immortal, was discovered in 1990s. That thing which I discussed earlier in the beginning. To understand what really happened that day, we must basically take a look to the life cycle of Teratopsis dohrnii, and simultaneously the life cycle of a common Hydrozoa, which is let's say Teratopsis rubra for the sake. So now guys, just take a look at these two life cycles on the screen. So first of all, you can see this one on the left is the life cycle of the Teratopsis rubra, and this one on the right is the life cycle of Teratopsis dohrnii. So in the initial stage, it's a medusa form, a reproductive medusa form, and this is also a reproductive medusa form in the case of Teratopsis dohrnii, right? So basically, if you just see it, so this is identical structure. If you just see this reproductive medusa and this reproductive medusa looks kind of identical. So as you can see, this is the dome-like structure, and these are the tentacles, and this is also a dome, and these are the tentacles, right? A dome, a basic structure, is a dome-like structure, and the tentacles and all that stuff, right? And these are the same, and these are the same kind of, right? These just looks kind of identical. So yeah, you can see these gametes right here. So that means this reproductive medusa just spawns gametes, right? This is the sperm, and this is the ovum, and then this just fuse and they become a planula. As I just told you, that the fertilized egg, that a kind of fertilized egg that is formed because of the fertilization of the reproductive gametes of the reproductive medusa, is known as a planula larva stage, like larval planula stage. So we can just say it a planula larva. So basically this same happens here also. These are the gametes and they just forms a planula larva, right? So this planula larva then just settles down in the sea, and it just just settles down, and then it just matures into a polyp form, which is a plant-like form. You can see, as I told you, this is kind of a plant-like form. It is an animal, but it's kind of a plant-like stage. If I just compare the life cycle of a butterfly simultaneously with it, my bad. So yeah, we can just see that if this is a adult medusa form and this is adult medusa form, then it's there is an adult butterfly form also, right? There is an adult butterfly form, and then this butterfly also produce gametes, and then that becomes into a fertilized egg of the butterfly in the leaves and the stems of the trees, and then that butterfly forms into like this form is kind of a pupa form of the butterfly, right? Like not even a pupa form, it's a caterpillar stage of the butterfly, which then turns into a cocoon, and there's a colony of the polyps. The colony of polyps is known as a hydroid stage, and this is the reason why we say these two hydrozoa, like they belong to hydrozoa class, because they spend their most of the life in this hydroid stage, whether it be the polyp stage or this, but the most of the life is spent in this, because in a like typical Pterodopsis hydrozoa, it just basically becomes just basically dies after all this stage. The maximum portion is in this part, right? But in the case of Pterodopsis donai, there's a catch, as you know this, because it's immortal. And why it's immortal? Because of this stage right here. This all happens because of this reduction phase. It is immortal, and this is the reason why it's immortal. Basically, this is the extra part or extra phase of its life. Then this Pterodopsis rubra, because you can see there is no such phase, right, in the life cycle. So there is a pure difference. You can just see it. It's just it can be just seen so naturally. So now you can just see here it's a planula larva, which just when the gametes fuses, then it becomes a planula larva. Here is also a planula larva, and this just becomes a polyp form, right? A polyp form. Here is also a polyp form. Just like forgot about this for right now, right? Just forgot about this. Just focus on this. This is a polyp form. This is a polyp form. This cyst just goes into the like ocean floor, and there's just planula form just that rises kind of, right? And there also the cyst just not cyst, my bad. This planula just settles at the bottom of the seas and just flows till it doesn't settles and like finds a starting place, which is like it can adapt to it, and it's safe right there. So it then just grows into planula polyp form, and these polyps then just like combine and forms a colony of polyps with medusa birds. So yeah, so you can just see this is a hydroid colony. This is a hydroid, one hydroid kind of thing, because it's a colony of polyps. So basically when polyps combine, then they become a hydroid form, and these small structures, you can see these small structures right here, bulges right here. So these are the medusa birds, right? Then these and then this hydroid colony produce a Genevilla medusa form, which is a younger medusa, like a pre-adult medusa, which just grows into an adult medusa, which is sexually mature. As we say immature medusa, then we are saying this Genevilla medusa form. Yeah, sorry, my bad. This form, this Genevilla medusa form we are talking about, because it's a kind of adolescent Pterodopsis rubra right here. So and these gametes just basically becomes planula larva, and that becomes a polyp, and then it becomes a hydroid colony, which is a group of polyps, and then there is a younger medusa form, and then there is a Genevilla medusa, which is a adolescent medusa form, and then there is a reproductive medusa, which is sexually matured, and this life cycle goes on. And a typical Pterodopsis rubra, from here this dies. The adult medusa eventually dies, right? But right here, if we just see, in every phase of the Pterodopsis donai, it can reverse itself back through the process called transdifferentiation. This magic trick is known as transdifferentiation, and it just like it reverses its aging process and it never dies. And when this happens is like when does this happen? When this transdifferentiation happens in this jellyfish is when it is triggered by any type of shock, a bad vibe, or it is just shocked or a danger or something, then it just reverses its its aging process. And when there if and a point to be noted, this kind of this shock can be really absurd also, because at the starting when there was one of the first experiment which is performed with this life form, then the scientist just uses a bit of tweezers right here. They just stretched its tentacles, and you know what, it just reverses its life cycle. And now if I just tell about this reduction phase, which is the main part, and I just told you to just forget about it, but now we're talking about it. So this is the reduction phase. So what happens is when it is triggered, then it just like its tentacles retract and it just becomes smaller and transdifferentiation happens. I'm gonna tell about what basically is transdifferentiation in depth. So this is it. So now this this reproductive medusa form, it becomes it just becomes a blob, and this blob becomes more smaller, and this blob like structure is known as cyst, and this cyst with early stolon. So this is just a cyst with a kind of a bulb like structure, and we can say, and then this bulb becomes a bud right here, a polyp bud. So a point to be noted is that planula larva is that when planula larva just grows into a polyp form, then it is a baby polyp. But on the other hand, when this cyst forms into a polyp form, then this is known as a reversed polyp form, because it's reversing. That is it. That's the main life cycle of this Pterodopsis dohrnii and Pterodopsis rubra, and that's the difference. You can see the point is this part is what we have to mainly study about, right? So what basically is transdifferentiation? So transdifferentiation is basically a process when a cell of one type just skips all the middle steps and transforms into a cell of another type, a really different cell, a totally different cell, without going to the middle stages and the middle steps. Basically a no detour from a generic neutral form, but is a total straight jump. And you know what is wild about Pterodopsis dohrnii? That it also doesn't have any stem cell. So stem cells are those basically we can do anything cells which can change into any tissue. And you know what that means? Basically the jellyfish is abandoning its cells old job and just forcing them to take a new job, a brand new job, and repurposing its cells. And that's literally so fascinating about this jellyfish. Scientists have tracked this down in the specialized areas of Medusa's body. Basically, it's outer dome first of all, which is an umbrella-like structure in the jellyfish, Pterodopsis dohrnii, and the second is the canal system, which is basically the gut. Why we chose these cells is because these cells are highly specialized for the Medusa's way of life. Because Medusa is the sexually matured part, so these are the most specialized cells in that Medusa. The dome cells are just built for swimming and structure, and the canal cells, the gut is basically, of course, it's for digestion. But when this jellyfish decides to reverse its life, then these Medusa cells stop being Medusa cells and starts becoming the new polyp cells. An entirely different toolkit for an entirely different body plan. This beautiful phenomena literally shatters biology's most oldest assumptions that once an organism reaches its sexual maturity, then there is no going back. In almost every other creature, once a cell locks into a particular job, then they're just stuck there until they die. But Pterodopsis dohrnii laughs into the face of that rule. It can basically rewind itself into every stage of its life, from a fresh young Medusa to a Medusa which is old enough and is literally on death's doorsteps. But as long as it gets the right trigger, the right shock to trigger this cycle. And sometimes that shock can be absurdly small. In the first lab test, scientists found that a little shock or a little, stretch from a tweezers is basically fine or appropriate for the jellyfish to just send it into its earlier life form. That's so hilarious. From an adult, then back into a blob, and all the way to a polyp stage. So now, basically, how does T. dohrnii prepares itself for its new life cycle. Like, is there any difference between the cells of cyst and the polyp cells? When scientists took a closer look to the cyst form of Ceratopsis doni, it was not found that it was just sitting over there waiting for its next phase. It was busy in ways which might explain its so-called immortality. While in the cyst form, the jellyfish diverts its most of the energy towards internal upkeep, especially protecting and repairing DNA. Its main target, telomere. Telomeres are basically the stretches of the DNA at the end of our chromosomes. Think of them like the plastic tips of the shoelaces. The preventer laces our DNA from fraying during cell replication. Every time a cell divides, a little bit of the telomere is shaved off. In most of the organisms, including humans, it wears them down, shaves down, and just becomes nothing. And as a result, these shoelaces, the laces that I think they are just removed, then this DNA is exposed to all the damages and danger, right? And that's the point when the cell starts dying, and so does the organisms. This shortening of telomeres is one of the key drivers of aging. But T. doni seems to have a countermeasure. In a cyst state, scientists have found an unusually high activity of the genes that produce an enzyme called telomerase. This telomerase enzyme produces telomere. So basically, these telomeres are kind of laces in the corners of the chromosomes, and they just prevent it from damage and aging, like shoelaces in a shoe does that for prevents fraying. This is the same rule of these telomeres in a chromosome. And as I said, these telomerase enzyme produce and increases the growth of these telomeres, and by expanding these telomeres, we can protect this DNA from the clockwork cycle. We can just protect it and prevent it from the clockwork cycle of death and aging. And guess what? That's not all. In its cyst form, it literally stops and gets a halt in the cell replication process and differentiation. That basically means it's not allowing its cells to break itself out or wear itself out or specialize itself out. The cyst that is formed doesn't literally even respond to external triggers and stimuli. It's like as if the jellyfish has locked itself into a high security vault, which is just using all its resources for repair and maintenance, and not growth and reproduction. Until the jellyfish is reverse jellyfish is well enough, that cyst form is well enough and grows enough, not even repairs enough to continue its life as a polyp. And that strategy clearly works. The T. doni is just spreading all over the oceans. And this is also giving an arising questions from the scientists. We have literally known this species from 1883, but we got to know about its immortality in 1990s, which is just so close. And now we suspect it's not alone. Like that, slob meme that arise that is immortal. Actually it's not. But we suspect that it's not alone. From fungi or trees, they just can have their own different, time travel cycle. And that's just so fascinating with biology, and that's what we are working towards, researching new things. Now, you must be thinking, does it never dies then? This life cycle can be repeated in imperfect conditions. A typical Ceratopsis doni might never die because of old age. But even though T. doni may bend the rules to regenerate itself, but it can't always cheat death. For example, jellyfish, including immortal ones, are also prey to animals, such as fishes, turtles, and polyps, whether it be reverse or normal, are also practically defenseless against sea slugs and crustaceans. Understanding how much time these jellyfish like T. doni can live can be tricky. T. doni is very sensitive, making it difficult to rear it in the lab itself. But guess what? Despite all these challenges, one scientist has been known to have long-term success in this captive immortal jellyfish. Meet Japanese scientist Shin Kubota, who has kept the populations of the immortal jellyfish looping back and forth from its life cycle from 1990s. Of course, his work with the species is time consuming, with him needing to monitor and take care of the species, the Tautopsis dorni, and also even breaking their miniature brine shrimp eggs, like literally small enough so that these miniature tiny jellyfish can eat it. But through his endeavors, Kubata has reported over a period of 2 years, the captive colonies of the jellyfish regenerated itself literally 10 times, and in many cases about just in an interval of a month. So guys, now we are gonna read a detective story which is written in jeans. So basically, I'm just gonna I'm not gonna read the whole research paper, but I have some notes right here which I made. The paper we're explaining is Matsumoto and Mijilita. It's written over there. 2021. The thing is, basically, we are just gonna read this research paper because in order to understand the how does this even happens, what does happens, and the deep the cellular level things and all that stuff. So it's gonna be fun. They just asked a simple and explosive question right here, what genes are switched on and off when the immortal jellyfish Turtopsis dorni reverses back from its adult form to its baby form. You're gonna understand this in few minutes as I'm gonna explain this. So to answer these questions, the scientists basically took the specimen of the immortal jellyfish in 4 stages. The first one is the polyp stage, when it is a plant-like thing, which is fixed and is anchored in the sea, in the seabed or something like that. The second, they took a medusa form, which is the adult form, which is swimming around the ocean and is capable to reproduce and spawn sperms and the ovum. And they also took the cyst stage, when it's like a blob-like structure, which is going to be which is going to be settled down and be anchored and then grow into a reverse polyp. And lastly, yes, they took the reverse polyp stage, which is a polyp which is formed by the development of cyst during the reductive phase, right? And after collecting all these samples, they just read the jellyfish to do by its RNA. Why RNA? I'm just gonna explain this. So basically right here, RNA are short-lived messages which your cells make when they're actively using a gene. So basically just think of it as a snapshot of what the animal is doing right now by measuring the RNA, right? So once more to revise, it's a polyp form which becomes a medusa form, and then that medusa reverses and becomes a cyst in its reduction phase, and that cyst becomes makes a reverse polyp. Polyp, medusa, cyst, and reverse polyp. And with this, all these specimens and the RNA measurement, they just made a transcriptome. Transcriptome is basically a huge list. It is like a inventory of RNA messages. So they just use this transcriptome to find out in which stages these messages are higher and lower to find the state specific programs, because in a stage where this particular transcript is higher, then its main functions to do that. So this just gives us an idea, and it's the central plot of this research paper, and it helps us to know which genes are switched on and off during a specific stage in the life cycle of this Turtopsis dorni. Basically this is it. So you're probably thinking, how did they actually do that? So basically they collected samples of T dorni from varied life stages, and they extracted its RNA, that is the active messages, and resembled and sequenced it. So basically they collected many small fragments of RNA sequences, read them and resembled them to a full-fledged message. And then they ran a differential gene expression analysis. That's basically a statistical comparison that asks which gene are meaningfully more active in one stage than another. The software used for this kind of work typically records a log2 fold change, which is basically how big the difference is. For example, if a log2 fold is equal to 1, then it means 2 times higher. An adjusted p-value or FDR, a corrected probability test, that tells you whether the difference is actually real or it's a random noise. When you test thousands of genes, if a gene has high fold value and a small p-value, then we generally believe or trust that the gene is actually different between stages. For the part like how these stats literally work in practice, the author used standard RNA sequences and tools like D sequence 2, and basically it was all standard. So now here's a one line from the paper. The cyst is a repair and maintenance hub. The medusa is an adult action hub with just swimming, sensing, and muscle contraction. And the reverse polyp turns on rebuilding and early development programs. So basically this means the cyst is a repair and maintenance hub. That means the genes which are for repairants, because cyst is actually becoming through the reverse polyp form. So it's reversing, it's repairing, and it's a maintenance hub because it is maintaining its overall structure and everything. And the medusa is an adult action hub because it's literally action, like performing action and mobility, because it's like swimming or sensing or reproducing, replicating. So it must have that power and ability to do that. So the genes required for that thing are more active and switched on at that period of time when the T-donai is in the medusa form, the adult medusa form. And the reverse polyp turns on rebuilding and early development programs because it's rebuilding, and it's early development program because the cyst is becoming the reverse polyp, and early development program because it's starting its life from the scratch once more, and it's just developing to the adult medusa form. That's what this basically means. So now let's important one of the most important chapter over here, the cyst. The authors found that when the jellyfish is in its cyst stage, then it turns on its lots of the genes which are perform DNA repair, telomerase activity, suppression of transposable elements, and ubiquitin related protein recycling. So basically ubiquitin gives the shape, so I'm gonna talk about it afterwards. So now I'm just gonna explain all of these paths, like why does the lot of genes are switched on in the cyst stage for these particular functions. So first of all is DNA repair. So basically in this portion the cell is basically like repairing damaged portions or the portions that are in danger or broken, like basically a repair crew who is patching potholes. So now telomerase activity. So telomerase is basically an enzyme which is used to increase the production of telomere. Telomeres are the protective cap-like structures at the end of the chromosomes, right? Like a shoelace, laces. Then there's a plastic cap in the end of the shoelaces which prevent the shoes to fray. In the same way, it also protects the chromosomes to be damaged or driven out. When telomeres get too short, then cells can't divide anymore, right? With more ease, because when they're nothing, when the telomeres just decrease to nothing, then the cell can't divide, and the cell dies because DNA is exposed, and as a result the organisms also die. So the enzyme telomerase can helps in production of the telomeres, which can increase or expand the telomeres on the chromosomes, and this protects from the aging process, and that's the why the jellyfish doesn't die. Now, suppression of transposable elements. The transposable elements are basically the elements that can move or can transpose from the genome and can often cause trouble if uncontrolled. So basically turns on the system, jellyfish turns on the system to keep them quiet so it doesn't trouble it. Ubiquitin related processes are like cells, trash tagging systems and recycling systems. The damaged proteins get tagged and broken down so they just don't cause trouble and damage to the. Now, why the cleaning even matters? Because the jellyfish does not appear to use the stem cells for reversal. Transdifferentiation is the name which is given to this process when a truly specialized cell turns into a less specialized cell or a totally different specialized cell without turning back to the neutral stem cell phase. Because the medusa cells are specialized for swimming, muscle contraction, and mobility. They generally don't turn into polyp cells unless you repair and reprogram them. So we can say that the cyst position over here is basically to make an environment which is safe or a safe condition to let the process of transdifferentiation to take place and happen. Now the Medusa, the adult jelly. So the Medusa stage basically shows the high activity of genes for membrane transport, like moving the components and substances in and out of the cell. Mesoglial components that provides buoyancy, and it's a middle portion of the cell that's kind of jelly-like, and it provides buoyancy and structure. The nervous system function and the muscle contraction. So, put it simply, the Medusa switches on its gene for feeding, sensing the environment, or swimming. At the same time, it suppresses the genes for early development stages or not even repairing, for growth and development. Because its main focus is on adult life, not regeneration, right? Now we're going to talk about something that is really important. So, when the cyst stops repairing and it is repaired, the animal reforms a polyp. But this reverse polyp is very different from a typical polyp in its gene activity. This reverse polyp is rich in genes which are involved in chromatin remodeling. Like, it's basically changing how DNA is packed and structured, and this would basically help us to switch on or off different genes, like different pieces. So simply you can picture it as like a group of group of string, like kind of a yarn. When we just lose one of the string, then we can just pull out any piece, and it just changes its structure and how it is packed. And matrix metalloproteinases, which is MMPs, these genes actually just remove the extracellular matrix, and it's like just removing the scaffoldings from a yarn so that new structures can be formed. And also the embryonic development programs, which are the genes which are produced when an animal is in its early development stages and it is growing and developing. It's not rebuilding, it's actually building. And actually, this combination is exactly what you would expect when an animal is rebuilding and just starting from the scratch. Look, here's the proof that the genes of the animal change frequently according to its basic needs at a particular stage in its life. When it's swimming, muscles and the nerves genes are dominating. When it's anchored at a place, then the feeding and colony genes are dominated. And when it's reversing, then repair and stress genes dominate and come up. With this, we have basically discovered how of the immortal jellyfish. We have to say that the immortal jellyfish genetic traits are not just cool, they are literally biological cheat codes. But here's the fun question that most of the people have: where on the earth are you gonna find this type of thing? Well, the immortal jellyfish was born buoy into the Mediterranean Sea, but now, thanks to us humans, it has literally backpacked its way throughout the oceans in the world. How? Cargo ships, cruise ships, it's basically hitchhiking for free. See, ships basically pull out, in and out, ballast water to keep its balance, right? So yeah, you just got it. This jellyfish just slips in and I am also coming with you. And then it just survives the whole ocean trips because when life gets tough, no food, bad vibes, it just reverses its life cycle. Duh. Like the ultimate, no, I'm just going back to my childhood move. Like literally. Oh, and really, it's the master of stealth. Tiny, see-through, and literally changes its fashion. With respect to the climate, in tropical spots like Panama, it literally just rocks in 8 tentacles. In cooler waters like Mediterranean or Japan, we can see it in 23 plus tentacles. Why? We don't know, maybe it's just a fashion thing. The hill hair part is, it's not even causing any trouble. No literally zebra muzzle like chaos. No Colombian hipporama. Yeah, it is also a thing. It's just out there vibing and casually being immortal. Of course. Anyways, that's the end of this deep dive in. And thanks for sticking around. And stay curious, stay weird. And with this, the Geek Goddess signs off. Bye guys.